Maybe you are not familiar with Corynebacterium, but you must be familiar with a species name called diphtheria. And diphtheria is one of this genus and one of the most important pathogen people worked on. And Dr. Boring discovered the existence of antitoxin against diphtheria, and he was awarded the first Nobel Prize in medicine, physiology and medicine. Okay. The full name of this diphtheria is Corynebacterium diphtheria. Name in Chinese. Bacteria looks irregular. So because there is some irregular swelling at one end, and for causing the bacteria look like club shaped, like this. This one we call it club shaped. So it's thicker on one end than the other, like this. Club shaped. If you stain the bacteria, you can see some granules stained deeply inside the bacteria. Those granules, just to remind that these granules are not spores, okay? These are not spores. They are just some material in it, some substance in it. This can help the bacteria to store energy. Granules in the bacteria. And when you make a smear and check it with a light microscope, you can see that the Corynebacterium usually they stay in parallel or at acute angles. So they like in parallel like this. So they are parallel lines, or at like acute angles like this. So that's because of some, I think it's the bacterial surface properties. So they, there is some forces between the bacterial cells. Okay, so this is morphology and identification. And they grow aerobically on most ordinary media. So this one is not anaerobic bacteria, it's aerobic bacteria. They prefer the existence of oxygen. So on agar containing potassium tellurite, this is the one salt of tellurite salt. The colonies are brown to black, and usually with a brown or black halo around the colony. That's because on this special, on medium containing this special chemical, they can produce some black material, and those black material is diffusible. And for diphtheria strains or subspecies, some of them are non-toxigenic, while some are toxigenic. And the non-toxigenic diphtheria becomes toxigenic when infected with bacteriophage from certain toxigenic diphtheria bacilli. And keep in mind that once the bacteriophage infects the toxigenic bacteria, then it infects the non-toxigenic bacteria. The non-toxigenic will become toxigenic. So that means that the bacteriophage bring some gene or some factor from the toxigenic one to the latter one. And this we learned before. That's the lysogenic conversion. So the bacteriophage form the prophage and bring some gene from the toxigenic one to the non-toxigenic one. Okay, so For pathogenesis, the bacteria usually spread by droplets or by direct contact, especially in kids. So that means that if there is some kid infected, we need to isolate the patient. The toxigenic diphtheria are capable of elaborating diphtheria toxin. And this diphtheria toxin is also an exotoxin, so it's a secreted protein. And it also contains two subunits. One is called subunit A, or we say that fragment A. Subunit A, or subunit A. And let's see how this toxin acts on the host. And again, the fragment B is the subunit binds to the receptor on host cell membrane, like this. The B binds to a receptor on the membrane. Well, after binding, the toxin is internalized and subunit A is released. The subunit A can inactivate elongation factor 2 and can prevent protein synthesis by ribosomes. And if the cell cannot produce proteins, cannot translate proteins, the cell will die. So it will cause a cell death. This is how the diphtheria toxin acts to host. And if there are a large number of cells die, it will cause a necrotic epithelium, and including like fibrin and red blood cells and white cells, and those dead cells will cluster together, will stick together by fibrin, and this will form like a membrane-like structure. A membrane-like structure we call the pseudomembrane, and it is this pseudomembrane which causes the obstruction of the airway. So it is this pseudomembrane which killed a person, because the person cannot breathe, cannot get the air into the lung. For clinical findings, at the beginning there might be only some sore throat or low-grade fever, sore throat or low-grade fever. And with the development of the disease, the patient might go through prostration or dyspnea. So that is the obstruction caused by the pseudomembrane, or suffocation, because they cannot get oxygen into the body. And from outside, you will see the swollen neck gland, and difficulty. The patient might suffer from difficulties with vision, speech, swallowing, or movement of arms or legs. So these are the different clinical findings. Here is the one famous example of diphtheria. This is a boy with severe diphtheria infection. And you can see the swelling neck lines, and also develop severe myocarditis, bone neck. Yeah, so this is diphtheria. And just now we mentioned a little bit about the history of diphtheria. We'll talk about that a little bit later, how to treat the disease. For diagnostic test, we collect specimens using swabs from the nose, throat, or other suspected lesions, and there might contains some bacteria. And we make smear and stain it with alkaline methylene blue. So alkaline methylene blue or gram stain. And then check it with a light microscope. If you want to culture the bacteria, you can culture on blood agar plate or a selective medium containing the tellurite. That's what we mentioned. If there is a potassium tellurite, the bacteria will form the black colonies or brown colonies. To test the toxigenicity, that means you want to detect the existence of toxin, you can use either ELISA or PCR to detect the toxin gene, or ELAC test. ELAC test is shown in the next slides like this. What you do is that you place a filter paper containing antitoxin this way, and then pour the agar into the dish. This is a culture dish. You plate the filter paper at the bottom, and then you pour in the agar to form a culture plate. Then you streak the bacteria this way. So there is a cross of the bacteria with the antibody, antitoxin. If the bacteria can produce toxin, the toxin will react with the antitoxin. You will see the precipitation line. So this line is the complex of antibody and toxin. So we call this a precipitation line. And you can actually see the line of precipitation. So you know that this line of bacteria can produce toxin, while this line is a negative control which cannot produce toxin. So you will not see a precipitation line. While this one might be the unknown one from your patient, and you can also see the precipitation line. So this way is called ELAC test. Through the ELAC test, you can determine whether there is toxin produced or not. For diphtheria treatment, we can give your patient some antimicrobial drugs, including antibiotics such as penicillin, erythromycin are the first-line antibiotics. And for like early administration or immediate treatment, you need to give your patient some specific antitoxin. That's the antibody. Usually it's animal serum containing antibody. You immunize a horse or other animal. You immunize a horse with diphtheria toxin and collect the serum containing antibody to give that antibody, anti-serum, to your patient. But a skin test must be taken for the patient to rule out the hypersensitivity to animal serum. So you don't want to bring in more dangerous factors to your patient. So this is early administration of antitoxin. Prevention and control. We can use active immunization. This one we introduced before. The kids will get a vaccine containing DPT, and in this vaccine it contains 3 different toxoids, and the D stands for the diphtheria toxoid. Okay, and the patients with diphtheria should be isolated because the disease can spread from person to person. So you need to isolate your patient and avoid contacting of your patient to other persons. Avoid the spreading. It's prevention and control. shake test is another test to evaluate whether an individual is immunized or not. So that we can ascertain the population risk. So this is carried out like this. You inject a little amount of antigen underneath the skin. If it's positive reaction, like this, reaction to antigen, that means that this person does not contain antibody to neutralize the toxin, to neutralize the antigen. So this person is not immunized, is not protected. So he or she requires immunization. And if it's a negative reaction, like this, there's no erythema on any arm, indicating that the toxin has been neutralized by the antitoxin, and the person is immune to diphtheria. So this is a shake test to see whether an individual is protected or not. I guess you can find this similar introduction in our textbook. Okay, so yeah, so that's all for this topic, this Corynebacterium diphtheria.